Hi everyone, I am Michaela. I will be presenting with Pranavi. Our presentation is on amyotrophic lateral sclerosis. ALS, also known as Lou Gehrig's disease, after the famous baseball player who was diagnosed with the illness, is a condition that attacks the nervous system, weakening the muscles and causing a drastic decline in physical function. The disease is interesting to science because how the disease is contracted is unknown. It was only 20 years ago that the first gene associated with ALS was discovered. However, it has been more commonly linked to veterans who have served in war and in people aging from 40 to 70 years old. Although there is no cure for ALS, there are treatments. ALS is a disease that is always fatal. At least 50% of people diagnosed with ALS will survive at least 3 years. 20% will survive at least 5 years, and 5% will survive at least 20 years. This disease causes the motor neurons in your body to die. Your motor neurons control body movement and reach from the brain to the spinal cord and from the spinal cord to the muscles. As the disease progresses, the affected individual loses the ability to walk, talk, eat, and eventually breathe. There are two types of ALS, familial and sporadic. Familial means it's genetically inherited. This type only typically affects about 5 to 10% of those diagnosed. The other is sporadic, and this means that the cause is unknown. This type affects 90 to 95% of the diagnosed. The symptoms of ALS vary. Muscle weakness is a characteristic in all cases of ALS, but people with the condition may also experience muscle cramps, clumsiness, slurred speech, twitching, and fatigue.
A buildup of aggregated proteins is common to both familial and sporadic ALS. ALS mutations were found in the genes SOD1, TARDBP, FUS, and C9orf72. The C9orf72 is the most common gene that causes ALS. The C9orf72 gene accounts for 10 to 15% of all the cases. The SOD1 gene counts for 2% of all ALS cases. The TARDBP and FUS are less than 1% of all cases. There have been numerous experiments done to try and determine the cause of ALS. One experiment found that the loss of motor neurons does not occur in the same manner for all motor neurons. There are two types of motor neurons which are affected by ALS. The first is the spinal motor neurons, which is categorized by SPMNS. The other type is the cranial motor neuron, known as the CRMNS. The SPMNS gradually determines which affects movement. The CRMNS, which affects the eye, are usually not affected. The experiment investigated why the spinal motor neurons and cranial motor neurons acted differently in ALS. In this experiment, the researchers used direct programming strategies and mouse modeling to investigate the differences. By exaggerating the HSOD1 proteins in vitro, induced cranial motor neurons and induced spinal motor neurons were created. Over time, less insoluble P62 was found in the induced cranial motor neurons than the induced spinal motor neurons.
Figure 1 presents the data and results of the induced spinal motor neurons and the induced cranial motor neurons. The researchers introduced chemicals in the experiment, which had resulted in the induced cranial motor neurons being more resilient than the induced spinal motor neurons. As mentioned before, ALS occurs when the motor neurons stop working. This has been associated with the depletion of TDP-43. Scientists conducted experiments on the TDP-43 at the molecular level. The scientists had knocked down the TDP-43 gene coding using a neuroblastoma, which is a cancer. The results had showed the reduction in the protein level. Although it was not immediate, it had finally caused the death of the cells. U6, which is one type of small nuclear RNA, decreased during the knockdown of the TDP-43. The other types of snRNAs were not affected. The scientists went on to add U6 into the depleted TDP-43 cells. The TDP-43 had stabilized the U6 molecules by binding to them. They had concluded that the TDP-43 did not directly cause the death of the motor neurons, but many sequences are.
There are various model systems which have been developed to determine the possible causes of ALS. Figure 2 shows the various model systems in ALS research along with the results. The scientists have found that the cellular models somewhat represent ALS. This helps in determining which genes are involved in causing ALS. Studying ALS by using rodents such as mice is considered the best option. Rodents have a physiology similar to humans. Other small animals such as the zebrafish, the C. elegans, and fruit flies offer the ability to test more than one hypothesis at a time. It is less expensive to maintain the other small animals as opposed to the rodents. Another issue with rodent testing, besides the expense, is inbreeding. The results of the experiments discussed so far will be presented next. The researchers had found in their experiments in the ALS modeling that when using embryonic stem cells and in vitro distinction, the induced cranial motor neurons and the spinal motor neurons degrade differently. The induced cranial motor neurons depend on its proteasome function to reduce misfolded proteins. The researchers concluded that the combination of in vitro and embryonic stem cells assisted in identifying the underlying distinctions between the cranial motor neurons opposition proteostatic stress.
Another experiment used to assist in finding the cause of ALS used TDP-43. The scientists discovered that the TDP-43 attaches to U6 siRNA, which stabilizes the molecules. The results show that the U6 siRNA was emitted as the TDP-43 was diminished. This may cause the death of the motor neurons. Further experiments show that the diminished TDP-43 could be restored to a point by the U6 siRNA. This suggests that the neurons could be rescued from dying, leading to a possible cure for ALS.
A great deal of progress has been made in the study of the causes of ALS throughout the years. There is now a better interpretation of the genetics of ALS along with the neuropathology. Several different models have been developed to evaluate the genes associated with ALS. The numerous models used to understand the ALS-associated genes appear to collect into operational networks. These models, which aid in differentiating the pathways of the ALS disease, give better insight into the causes of ALS. There are many future directions resulting from each experiment. The researchers suggest a future study that could involve co-culturing the motor neurons with astrocytes to evaluate any position in ALS advancement using the embryonic stem cell platform. In the experiment that found the U6 snRNA aids in normalizing the TDP-43 and regenerating the motor neurons. Continued research should be done to evaluate the method and process. This experiment definitely shows promise into reversing the death of the motor neurons, but definitely needs further investigation into the mechanism to determine how the U6 snRNA actually helps the TDP-43. The future direction in ALS is to continue modeling this disease and continue the research to stop or at least slow down the death of the motor neurons. These are the references used in this research paper, and thank you for listening.
And that concludes our presentation. Thank you for watching.